Hi everybody. Let's have a deep dive into our recent study. Multi-omics reveals different signatures of obesity-prone and obesity-resistant mice. Obesity-prone (OP) and obesity-resistant (OR) mice exhibit distinct metabolic profiles. Under a high-fat diet, OP mice experience rapid weight gain, whereas OR mice show a marked resistance to obesity. Recent studies have highlighted that the gut microbiota significantly influences host metabolism, immune responses, and gut barrier integrity through metabolites such as bile acids, amino acids, and short-chain fatty acids. Therefore, investigating the role of host microbiota interactions in obesity phenotypes is crucial. In this study, we integrate microbiome, metabolomics, and transcriptomics to investigate obesity-related phenotypic differences and host-gut microbiota interactions in OP and OR mice. Our findings reveal notable differences in bile acid and amino acid metabolism, as well as distinct variations in key bacterial taxa, such as Longibaculum and Kineothrix, between the two groups. Moreover, Kineothrix, intestinomonas, and Forniella are enriched in both OR mice and OR human. Notably, non-12-OH bile acids, including ursodeoxycholic acid and hyaluronic acid, were significantly elevated in OR mice, helping mitigate high-fat diet-induced weight gain. Additionally, 22 specific amino acid profiles are identified as potential biomarkers of obesity susceptibility. To evaluate the high-fat diet's impact, we monitored body weight changes. By week 3, OP mice gained significantly more weight than OR mice, with divergence increasing over time. At week 5, microbiota analysis showed OP mice enriched in Longibaculum, while OR mice had Kineothrix, marking the first major microbial shift. By weeks 14 to 16, OP mice were dominated by Longibaculum and Agathobacter, whereas OR mice harbored Kineothrix and intestinomonas. qPCR confirmed these differences, with Kineothrix, intestinomonas, and Forniella consistently enriched in both OR mice and humans. What about the host metabolism in OP and OR mice? We conducted serum metabolomics and identified 1,567 metabolites with significant differences between the two groups. Pathway enrichment analysis revealed alterations in amino acid metabolism, bile acid regulation, and energy metabolism, indicating that metabolic reprogramming may play a crucial role in the phenotypic differences observed between OP and OR mice. Fecal metabolomics further confirmed differences in amino acid and bile acid metabolism between OP and OR mice. Specifically, non-12-OH bile acids were significantly elevated in OR mice, demonstrating potential anti-obesity and gut barrier-enhancing properties. These findings explored the interaction between metabolites and the microbiota and their impact on gut homeostasis and obesity susceptibility. Future research will further investigate the specific relationship between these interactions and obesity phenotypes. Do the phenotypic differences in obesity correlate with changes in gene expression? Clone transcriptomic analysis revealed that OP mice exhibited upregulation of genes associated with immune activation and gut structural damage, whereas OR mice showed upregulation of genes related to cell adhesion and gut barrier integrity. Key driver gene analysis identified 10 genes closely linked to gut immunity and barrier function, all of which were significantly upregulated in OR mice. To validate these findings, we performed IL-6 immunohistochemistry and found significantly elevated IL-6 expression in OP mice, indicating a heightened inflammatory response. qRT-PCR further confirmed the upregulation of 8 key driver genes in OR mice. Correlation analysis revealed complex interactions among gut microbiota, metabolites, and regulatory genes. Kineothrix correlated positively with bile acid metabolism, while Longibaculum showed an inverse trend. Bile acid metabolites were linked to gut barrier protective genes, whereas amino acid metabolites had the opposite effect. OP mice were enriched in amino acid metabolism pathways, while OR mice showed reduced enrichment in digestive system pathways. This study sheds light on the dynamic alterations in gut microbiota composition between OP and OR mice, identifying Longibaculum and Kineothrix as key bacterial taxa. We found that OR mice exhibited elevated levels of non-12-OH bile acids, which reduced high-fat diet-induced weight gain. Furthermore, 22 distinct amino acid profiles were identified as potential biomarkers of obesity susceptibility. In sum, host microbiota interactions may influence the differentiation of OP and OR phenotypes, providing important insights for a deeper understanding of the mechanisms underlying obesity susceptibility and resistance.